Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss nuclear DNA replication in humans. First, a quick overview. In a previous video, we discussed the cell cycle, which consists of various phases, G1, S, G2, and the mitosis phase M. During the G1 phase, the origin of replication in a human DNA, a human chromosome, and you can see there are several origins of replication in human DNA compared to E. coli that we discussed previously, that has only one origin of replication. So to each origin of replication, we have several proteins that will come and bind. These include original recognition complex, CDC6 protein, CDT1 protein, as well as helicase, which is the MCM27 proteins. So all of these proteins will bind to the origin of replications. And here we see helicase shown in blue binding to all the origins of replication. This is called origin licensing. Now, during the S phase, other proteins such as CDC45 and the genes complex will be attached to helicase to form CMG helicase. So we see this proteins here in orange, which is the CDC45 and the genes, attaching to helicases on some origins. So only on some origins, this one here, for example, this origin here, are these proteins attached to helicase. This one here, for example, doesn't have that. This doesn't have that. So CMG helicases that have these proteins will now become activated at these origins. The activation of CMG helicases at some of the origins is called origin firing. So origin licensing occurs in G1 phase, and origin firing at some origins occurs in the S phase. So wherever we have the activated CMG helicase, the helicases will unwind the two strands as shown here and as shown at this origin. And in the next step, other proteins will be recruited to the helicases to form replisomes, and these replisomes will copy the entire DNA to form two copies of each chromosome. So that's basically an overview of how human chromosomes are copied during the S phase or synthesis phase. A few more points before we get into the details of human nuclear DNA replication. In E. coli, DNA is found in the cytoplasm of the bacterium. In case of human, as we know, most of the DNA is found in nucleus, and in the mitochondria we do also have, each mitochondria also have anywhere between 2 to 10 circular DNA. This is called mitochondrial DNA, and this is circular. Nuclear DNA is linear, okay, while as we saw in the previous video, E. coli DNA is also circular. As far as origins of replication, there is only one origin of replication in E. coli. In a human nuclear DNA, there's a chromosome. There are several origins of replication, and later in the next video we'll look at the mitochondrial DNA replication. There we have two origins of replication. Okay, so let's look at the key steps in human nuclear DNA replication. During the early G1 phase, the concentration of cyclins is very low, And without the cyclins, or because of low concentration of cyclins, these CDKs, these kinases are not activated. And without kinase activity, most of the proteins present during this time are dephosphorylated proteins. The dephosphorylated active form of the origin recognition complex protein, ORC, will recognize the origins of replication and bind to it, and then the ORC will recruit the dephosphorylated CDC6. And in the next step, the ORC CD6, shown here, will now bind the double inactive MCM27, which is basically the helicase. So the helicase will now bind to the origin of replication, and this helicase has attached to it another protein called CDT1. And all of these binding forms the pre-replication complex, and this completes the origin licensing. Now, origin licensing occurs only in the G1 phase. As the cell cycle proceeds from G1 to S and the other phases, we can see various cyclins increasing in concentration. So after G1, the concentration of cyclins start increasing tremendously. And with high cyclin concentrations, the CDKs become active, and these CDKs will now start phosphorylating several proteins. For example, ORC, when it's phosphorylated, will inhibit complex formation with the helicase. CDC6, when it's phosphorylated, will become ubiquitinated and degraded by the proteasome. So this means there is no helicase loading or origin licensing after the G1 phase, that is during the S phase, there's no licensing. G2 phase, there's no licensing. M phase, there's no licensing. Only G1 phase is where we see the origins becoming licensed with the attachment of the pre-replication complex. So after the origins have been licensed during the S phase, as cyclin concentrations increase and phosphorylation happens, the phosphorylated ORC and CD6 will be removed from the origins. So we see these phosphorylated proteins being removed from the origins. During this time, during the S phase, there are kinases such as the DBF4 dependent kinase or DDK. This kinase will phosphorate helicase, while other kinases such as ECDK2 will phosphorate other proteins, and this allows recruitment of these two proteins here, CDC45 as well as the genes complex. It allows the recruitment of these proteins onto the MCM27 helicase, and this happens only on some origins. So these proteins are recruited to some origins. And the attachment of CDC45 and genes complex to MCM27, which is the helicase, will form functional CDC45 MCM27 genes protein that we call CMG. So this forms CMG helicase complex. So from now on, we'll call helicase as CMG helicase. So the CMG helicase complex is formed on some origins, and this is called origin firing. Now, although functional CMG helicase are formed on certain origins, these helicases are still inactive because there is still CDT1 bound to the helicase. CDT1 inhibits helicase activity. So at this stage, the helicase cannot unwind DNA at the replication fork. So in the next step, we have proteins such as the E3 ubiquitin lipase protein called CRL4-CDT2, and as well as several other proteins such as the SCF, SKP2. These proteins will initiate the ubiquination and degradation of this licensing factor CDT1. Besides degradation of CDT1, CDT1 can also be inhibited by a protein called geminin during this time, and by hyperphosphorylating CDT1, especially by the cyclin A CDK1, that can also lead to inhibition of CDT1 activity. So through all this means, CDT1 is removed, destroyed, or inhibited, and now the helicases are activated. They don't have CDT1 anymore, and these helicases will now begin unwinding the two strands of the DNA on some origins, not all origins. Once these two strands are separated, then we have these proteins called replicative protein A or RPA. They will bind the single strand, each single strand that has been unwound, and they will prevent the single strands from rejoining or re-annealing, as well as attacked by nucleases. Remember, in E. coli, this was done by single-stranded binding proteins. The DNA unwinding by CMG helicase will cause twisting of the DNA that is not separated, and this twisting will cause the DNA to wrap around itself to form positive supercoils. These positive supercoils that are ahead of the fork, right down here as well as here for example, will hinder CMG helicase from unwinding more DNA, and it will stop the replication process. A type 2 topoisomerase that is called topoisomerase 2 alpha will relax these positive supercoils back into their relaxed form, and this will now allow the CMG helicase to continue unwinding the DNA. Let's follow the events that happen at one replication fork. Clearly, the same events happen at the other replication forks. In the next step, recruitment of several other proteins will form the replisome. So examples of these proteins are DNA polymerase epsilon, Pol E, or Pol epsilon. This protein copies the leading strand, that's this protein here. Then we have DNA polymerase alpha, which is the primase or Pol alpha. This will synthesize RNA. So here is our Pol alpha. DNA polymerase delta or Pol delta, that will synthesize his Pol delta. That's the one responsible for synthesizing Okazaki fragments from the lagging strand. The protein RFC, which is replication factor C, is comparable to the clamp loader complex in E. coli. This protein will load clamps, and these clamps are called proliferating cell nuclear antigen or PCNA. That's comparable to the sliding clamp is in E. coli. Note that, and we'll talk about this in the next video, there is another polymerase called polymerase gamma or Pol gamma. This is used for mitochondrial DNA replication. Okay, let's look at copying the leading strand. The replication factor C, RFC, will load a clamp on PCNA clamp on the leading strand, and you can see that happens here. And then once the clamp is loaded, Paul epsilon will come and bind to the clamp. This PCNA will prevent polymerase epsilon from falling off the template strand, thereby increasing polymerase process sensitivity. Once Paul epsilon is attached to the leading strand, Paul alpha will come in and make a short RNA primer. The Paul epsilon will use the leading strand as template, and it will copy the leading strand as shown here. Remember, synthesis happens 5' to 3', and this newly formed strand is running anti-parallel to the leading strand, so this leading strand can be copied very easily. Just as in E. coli, now copying the lagging strand is a little bit more complex. So what happens, once again, Paul alpha will make the primer, an RNA primer that will provide the free 3' OH group necessary for DNA synthesis by DNA polymerase. The RFC, this protein here, will then load a clamp, PCNA, onto the lagging strand, and once the clamp is loaded, the DNA polymerase, in this case Paul delta, will come and bind the lagging strand. So the Paul delta will now extend using the 3' OH of the RNA primer. We'll start copying the lagging strand and will make new DNA as an Okazaki fragment. Note once again that the helicase is going here from left to right. The leading strand, 5', 3' synthesis is also going left to right. The synthesis of the copying of the lagging strand, 5' to 3', is going in the opposite direction from right to left. So after the Okazaki fragment is made, the Paul delta as well as the clamp PCNA will dissociate from the DNA. So as the CMG helicase keeps on moving, in this case from left to right, the newly synthesized DNA that was previously synthesized will pair with the parent strand as shown here. Similarly, the Okazaki fragment that was made earlier will pair with the lagging strand. So just as what we saw in E. coli, the leading strand is copied continuously. So this strand here is copied continuously, while the lagging strand will be copied as fragments, Okazaki fragments. Now replication is coordinated between the leading and lagging strand by looping the lagging strand. You can see the lagging strand here is being looped as the replisome moves along the DNA. Now this looping will bring the synthesis site closer to the replisome, the synthesis machinery, and this will allow both strands to be copied simultaneously despite opposite synthesis directions. Since this structure here resembles a trombone slide, this form of replication is sometimes called the trombone model of replication. So here we see making more Okazaki fragments. You can see a Paul alpha will make an RNA primer on the looped lagging strand to start a new Okazaki fragment. Once again, the RFC, replication factor C, will load a PCNA, and then the polymerase delta will come and bind to the pCNA, and it will start using the primer for its 3' OH, will start copying the lagging strand into a new Okazaki fragment. Now this polymerase delta has 3', 5' exonuclease activity, and this will help it displace the 5' end of the RNA primer on the downstream Okazaki fragment into a flap structure. So this fragment here, this RNA primer here, will become a flap structure. Another enzyme called flap endonuclease 1 or FEN1 will then cleave this RNA flap. Then the enzyme RNase H1 and H2, these are endonucleases, will now degrade this RNA flap into smaller RNA fragments. A similar mechanism might also occur here on the leading strand. Once the RNA primer is removed, then the pol delta will now use this 3' end to fill in the gap where RNA primer was destroyed. Okay, so this resulting gap here will be filled in by polymerase delta. Then another enzyme called DNA ligase will now join this Okazaki fragment with this Okazaki fragment. So it will join these two ends, and that will produce a single continuous strand when the Okazaki fragments are joined together by the ligase. Now, when the replication fork meets a nucleosome, obviously the nucleosome is going to obstruct the progress of the replication fork. So here we need the help of other proteins such as histone chaperones as well as chromatin remodelers, and these proteins will cause the disassembly of the nucleosome so that the replication fork can pass through and copy all this DNA that is wrapped around the nucleosome. And once the replication fork leaves the area, then these proteins will reassemble the nucleosome. So this process we just discussed will keep on repeating as the replisome moves along the DNA. It will copy both the leading strand as well as the lagging strand. Now, copying the ends of linear DNA, the ends of its chromosomes are regions called telomeres. This causes special replication problems. So let's look at a little bit about the structure of the telomere and what kind of problem we see when we try to copy the ends of the chromosome. The telomere has a T loop, which is basically a telomere loop, and you can see the telomere loop, the T loop here. And then there's a smaller triple-stranded D loop or displacement loop. You can see one strand here, another strand here, and then another strand here, forming what is called a displacement loop or D loop. Now the 5' end, shown here in purple, has several repeats of this CCC-TAA. Remember, we always have to write sequences from 5' to 3'. So from 5' to 3', this sequence here would be CCC-TAA. So this purple loop, this 5' loop strand, has a lot of this CCC-TAA repeats. Very rich in this CCC-TAA repeats. The 3' end, shown in brown, this has a lot of TTA-GGG repeats. TTA GGG repeats. A lot of these repeats on this strand shown in brown. The 3' end also has what we call a 3' overhang, or a G tail, which is about 75 to 300 nucleotides long, and you can see that tail here. You can see an extra piece. This is called the G tail. Okay, the 3' overhang, or the G tail. Now there are several proteins that are found on the telomere. For example, there are proteins called protection of telomeres 1, or POT1, shown in red here. And these proteins will bind these unpaired single-stranded TTA GGG repeats, these repeats. And this binding will protect this part from being recognized as a DNA break. Other proteins called TTA GGG repeat binding factors, or TRFs, shown in green here. These will bind the duplex repeats. So wherever there's this double-stranded repeats, these proteins will bind and protect it. So basically the telomere is a region that has loops, repeats, as well as several proteins that is protecting it. So let's look at the problem when we reach the end of the chromosome. This is called the end replication problem. So in the lagging strand, remember, we are making Okazaki fragments. So here we have these Okazaki fragments, they're already joined, and now we are ready to make another Okazaki fragment here. So when this last RNA primer is removed from the lagging strand and degraded, so when this RNA primer is removed and destroyed, we have a gap here. So there's going to be a gap here. Now, there is no Okazaki fragment down here, so that means there is no 3' OH present, which means Paul delta doesn't have a 3' OH. That means it cannot replace this RNA primer with new DNA. So it cannot fill in this gap. This means that every time the chromosome divides, there will be this gap present at the end, this copying of the lagging strand. And this gap would be at least the size of one RNA primer. Okay, so this would mean that the chromosome would progressively shorten after many cell divisions. So this problem is resolved by another enzyme called telomerase. Now telomerase is a ribonucleoprotein. It has an RNA template in the enzyme. So it's basically a RNA as well as a protein. That's why we call it a ribonucleoprotein. And this RNA template is CCC AAUCCC. It has that sequence. So the telomerase will bind the G tail of the telomere through its RNA template. So you can see the C here binds with a G, this C here binds with a G, here's another C binding to a G. So this way the enzyme will come and bind to this area. Now the telomerase will now use this RNA template to extend this tail, to extend the tail. So for example, you can see the A here has been made into a T at the end of the tail. This A here has been copied into a T to extend the tail. This U here has been copied into an A. The CCC has been copied into GGG. So since telomerase is making DNA from RNA, it is a reverse transcriptase enzyme. Okay, reverse transcriptase. That means it's making DNA using RNA as a template. So the telomerase will keep on extending this G tail many times, multiple times, using its RNA template. So with this extension now, the replisome can come into this point, make an RNA primer, and then synthesize DNA by polymerase delta and fill in this gap. So in this way, the replisome can now complete copying the lagging strand gap. So this is how one chromosome in the nucleus is copied to form two chromosomes, and this is what happens in the synthesis phase of the cell cycle. In the next video, we shall discuss mitochondrial DNA replication. So until next time, thank you very much for supporting this channel. Have a wonderful day, and we'll see you soon.